Okay, hello everyone. Welcome back to our Eric and Wendy Schmidt Center Colloquium, which is jointly hosted by MIT EECS and here the Broad. I'm very excited to have Olga here with us today. So as you know, in this colloquium series, we feature speakers who, on one hand, take machine learning methods into biology and try to and answer important questions in the biomedical space, and also the other way around, go and work on foundational machine learning developments spurred by biomedical questions. And so here is a speaker today who is like seamlessly going between, the machine learning world and the biomedical field. So it will be really exciting to see. So Olga is a professor in computer science at Princeton University and also in the Louis Sigler Institute for Integrative Genomics. And she is founder and director of the Princeton Precision Health, which I'm looking forward to learning even more about. And in addition, she's deputy director for genomics at the Center for Computational Biology at the Flatiron Institute of the Simons Foundation. So she's received many, many prizes, including the Sloan Research Fellowship, NSF Career Award, Overton Prize in Computational Biology, et cetera, et cetera. And I'm sure many of you already know her research. So she focuses on the development of machine learning methods to decode genomes. And does this really with a goal of diagnosing and treating complex diseases. And we all know her from a lot of her pioneering work on Bayesian networks Bayesian methods for networks in cellular tissue context, and thinking about regulation in health and disease. Also her work on looking at the non-coding genome and looking at that again in contexts of health and disease, and in addition, also integrating many different modalities in order to get like a systems level view or a systems level and molecular view of human health and disease. So we'll have a treat here today, and we'll be learning about deep learning models for genomes and precision health. So welcome, Olga. Very happy to have you here.
Thank you so much for such a generous introduction, and it's really fun to be in this audience that's I know is very much mixed and thinking about these machine learning and computational approaches to various problems in biology. So I wanted to just start by a very general motivation, which I'm sure is familiar to all of you. How do we really get to the point in the future where we could really potentially have a world where diagnosis, treatment, and even health recommendations can actually be based on some combination of the genome and various environmental and socioeconomic, as well as other types of factors that really affect your environment, right? So all of your clinical data, the environment you live in, your lifestyle information, etc. Basically, the goal is to really integrate all of this to be able to base diagnosis, treatment, and development of drugs and treatments as well from all of this combination, as well as, of course, with the goal of improving outcomes. So I'm going to focus today on the molecular side of this whole equation, really on the genomes, but I know many of you here are working both on the sort of molecular side of it, but also even starting to think about the environmental and condition-specific aspects of this. And so would be very happy to talk about that as well if you're interested in the questions. Obviously, we all know about the genome, and I'm going to bore you a little bit, many of you a little bit, about the basis of this. Of course, we all know about the ATCGs and the code, but what I really want to emphasize is that while we know a lot about the genome, and we know exactly where the coding regions are, we know a lot about the regulation, we actually still to this day don't really have a great read, or at least certainly far from a perfect read, for really the full code of what every variant in the genome means, right? And I especially want to really draw your attention to the non-coding space in the genome, right? So, of course, the vast majority of disease genetics up to about, I would say, 10 years ago or so has been largely focused on the coding variants and coding aspects of this, and they're extremely important. But in reality, of course, this is much more complex, right? So while it's really important to understand what's happening on the coding level, under the surface of this is, well, first of all, the huge diversity of the coding level, right? So all of the isoforms, the splicing, the transport, right? So even if you get that isoform, where is it actually going to go? Especially in cells like neurons, where it is a huge difference if something makes it to the end of that axon or it doesn't, right? So those, the huge diversity of isoforms that we see across cell types and tissues, all of that is, of course, regulated by non-coding variants that are affecting binding of RNA binding proteins in this case, as well as the transcriptional regulation, right? So the variants in the 98% of the genome outside of genes that are controlling either through proximal or through distal regulation how much of that particular gene product is produced. And so that's actually extremely important. And I just wanted to show you one example that was not something that we discovered. That was actually a paper that was published in 2006. And they're looking at a very well-studied disease called alpha thalassemia. So the sort of Mendelian form of alpha thalassemia is caused by, basically they studied this group of about 148, 150 individuals in, I'm blanking, I think it was Malaysia. And these individuals, it was very interesting. They could not find a single coding variant in this group. And then once they started looking very carefully, so the standard form, basically they don't have proper folding of alpha globulin, and that's how you get the disease, right? It turns out that in this case, they had perfectly functional alpha globulin coding region. What they had is, and here you can see and this is data from that original paper, right? A reference allele and a mutant allele. What you see is actually, there's a variant right here where what it's causing is an overexpression. You can see this intragenic expression that's happening in the mutant allele and not in the reference allele. And what's most likely happening is that this intragenic expression is repressing expression within one of the exons of this alpha globulin protein. So these individuals have a perfectly functional coding region, but effectively have a mutant protein and end up with a phenotype that is identical to a coding mutation. So we actually stumbled upon this example because after we developed our first approach to looking at transcriptional variants, this was one of the very strong predicted variants that was actually repressing expression of this particular protein, and we stumbled upon this paper. So now that I hopefully motivated to you that it's actually really important to look outside of genes, why is it hard to look outside of genes? Well, of course, this is hard because all of these individuals might have the same disease, but under the surface, right, the molecular basis of this disease could be very different. That's not news to any of you, I know, in this audience. But remember, now we're in the non-coding genome space. So this is 98% of 6 billion letters, right? Oh my God, 3 billion letters, right? So at some point, I will actually calculate how much that is, right? And at each point, we can have 4 different choices. So this is a huge search space, right? So, and this is why it's hard. So if we try to develop standard sort of quantitative genetics approaches by themselves, where we can look as just common variants, right? And then we have LD blocks. So this is very powerful, but we need a complementary way to do this that allows us to really study variants without limiting ourselves to only common variants. And ideally, what we really want to study is variants that we have never seen in populations yet, right? Matched to a phenotype. So what I'm going to tell you about is some of the methods that address these questions. So the sort of 3 questions I really want you to think about is, how do variants impact transcriptional and post-transcriptional gene regulation? Which variants are functional and affect cell type and tissue-specific gene expression, right? So we can have some variants that are actually not doing anything, really, right? Like you can have a change in the genome, it does nothing. But you also can have actually functional variants that are completely benign, right? Like there may be a group of functional variants that control the fact that my hair is curly and yours is straight-ish. All right, and there's others that might be disease causal, right? And so we want to be able to map all of that, and we want to be able to develop deep learning and AI-based methods that are really going to predict regulatory effect of variants in a way to basically mimic what genetic code has done for us in the coding space, right? So not having to actually have examples where we have a genome matched to a phenotype or genome matched to a molecular phenotype, but being able to really understand in the sequence without bias towards that variant's commonality or rarity, what is its regulatory effect. We want to understand that effect in a cell type as well as condition-specific ways. So, for example, what's happening early in the development and a very specific neuronal cell type. And we want to understand both transcriptional and post-transcriptional effects. The other part, of course, of this equation is that these variants are not going to act by themselves. They're acting in concert, right? And they're impacting these complicated cellular networks that are, again, highly context-specific. And so what we want to be able to do is also thinking about how can we understand these cellular networks to study dysregulation and disease with the goal of eventually bringing those two aspects together, right? So we can understand genomic effects in a context-specific way of how they impact and dysregulate these cellular networks. So what we really want to do is be able to look at any variant. So there is one there, right? This particular C, let's say, is a variant from a reference allele. And we want to have some sort of AI model that's going to say, well, if you have an A in a healthy individual and a C in disease, maybe that's preventing binding of a transcription factor. My experimentalist in my lab hates this slide because she always gets upset that the transcription factor, it does not disappear. You all know it doesn't disappear. It just doesn't bind. I'm also aware it doesn't disappear. I did get rid of the one where it blew up and fell into pieces. She was extremely unhappy about that one. So, all right. So here's what it actually looks like. This is one of our very early models. Now it's more complex. I'm going to sort of go between levels, right? Because there's, I know, some very technical people here. There's some biologists, I'm assuming, here. So I'm going to go between levels in this talk. I will mostly keep it high level, but I want to show you a couple of deep learning frameworks sort of under the hood. So this is sorry, I'm finding it really hard to use this. Look at this screen because I can barely see the screen. There we go. All right, so this is the structure. It's a CNN. I will show you the more recent model that's actually larger, but this actually gives a really good intuition of why these models work. So I know some of you probably build similar models. If you don't, here's the intuition. So the way quantitative genetics works, right? Like you have a genome and you have some output. So in this case, the output we're trying to predict is epigenetic regulation, right? So let's say a ChIP-seq experiment. So whether a transcription factor binds or not, ChIP-seq peaks or histone marks, et cetera. Like, let's just think about transcription factors because they're easier to talk about. So let's say we're trying to predict GATA1 binding. So we will have right? Like, so ideally in a quantitative genetic framework, I will have thousands of individuals sequenced or thousands of cells sequenced, and I will know for which ones there is binding and for which ones there is not, and I will build a statistical model. Now, remember, we're trying to do this without being able to then predict the effect of rare variants. So what we're going to do is instead, we're going to do a unsupervised version of this, except for it actually is kind of supervised. So for those of you computer scientists, this is kind of almost a philosophical question. So it's a non-supervised setup, but you could think of it as supervised because what you have is many, many examples of GATA1 binding within the same genome. So we don't actually use variant information at all to learn. We just use the reference. I mean, it doesn't have to be the reference genome. We just use the reference genome, but it could be any genome. So actually, that genome is not matched to any of the ChIP-seq experiments that we're training on, right? Because we know that's not from Craig Venter or whomever the reference genome came from, right? So the reason that the model is able to learn is it's sliding along, there's seeds here at the front, you guys. It's sliding along the genome, and it's seen many examples of GATA1 binding. And what it's able to do, even if that's not an exact match to those cell lines or tissues in which the GATA1 ChIP-seq was actually measured, there's enough examples that it's able to learn what is actually important in that signal. Furthermore, because it's a deep learner, right? So this is what this part is doing, right? So we're doing sort of motif scaling, and then it's basically spatially looking at motifs across like smaller motifs that it's integrating with. But then furthermore, remember, we're not just looking at GATA1 binding. We have many different features we're training on, right? So we have different histone mark, other transcription factors, et cetera. So the model is also able to pick up on higher order interactions between not just spatially, but also between different features. And that's another powerful part of what's giving it signal. And so at the end, this context integration is going to give us, basically it's going to train on a bunch of cheap seq data. And then when we're trying to go into the prediction state, we can give it. So that means it's going to say, OK, for this sequence, right, the probability of binding is very low. Whereas for this sequence, which is just a single nucleotide different, the probability of binding is very high, right? So that means that now it's learned actually the sequence, basically language of binding, without any statistical association. So it now can predict the effect of any variant, even ones that have never, ever been seen in any sequencing study. All right. And there it is. So what we actually are able to do then is take these models, and actually we go beyond purely predicting epigenetic and biochemical effects. So we can basically think of it as in silico cheap seq with these models, right? So you're able to predict these biochemical effects. Then you can actually take these and train them now in completely supervised way to predict pathogenicity. So saying, if we look at variants that have been documented, regulatory variants that have been documented in disease databases, we can learn what their biochemical and molecular profile looks like from predicting their effects in these models and compare them to every new variant. One other important part of this is that you actually get a specific mechanism, right? So you get a prediction not just of, oh, I think this variant is going to prevent GATA1 binding, whereas it should be binding in a particular, let's say, immune cell type in a particular context. You also can tell, and that if that specific event is going to cause overexpression of a specific gene in this specific cell type, right? So you get it, or it will affect splicing, for example. Obviously not GATA1 binding, but a particular variant could affect splicing. What I do want to point out is, and I can talk about this more if you guys are interested, this part of the model is actually highly unbiased. Like nothing is perfect, obviously, in biology. There's always biases. I do want to point out that in a lot of our pieces, we actually really focus on this, and we kind of, we'll show pathogenicity because that's what people really like in papers. But in reality, this part is very biased, right? Because this is where you have to look at the variants that have been already found. And it's just a very limited set of variants, comparatively, that has been in the regulatory space. Remember, this is non-coding. So there's just a lot smaller set of variants. And as you can imagine, they're highly biased, right? Because most of the regulatory biology has been sort of the easier to find things on promoters, very few enhancers, right? So it's a very specific set. So this works, but this step biases you a lot. So I just wanted to give you that intuition in general. This is not just for us, but for any such model, right? Like as soon as you look at pathogenicity, you really have to supervise with variants. All right. So this was just the general information of how these models work. So now I actually want to tell you about our current model that actually goes beyond this biochemical aspect of things and really starts thinking in terms of what is it actually doing in terms of regulatory activities. And the reason that we think about that is, so SAY actually can predict about 22,000 different features, and I'll tell you more about it. So 22,000 basically cheap seqs. Think about it that way, which is great. It's very powerful. But what does it mean, right? So you ended up, for every variant, a vector of 22,000 effects. It's great biochemically. But isn't it more useful if you can actually say what is it actually doing is it's disrupting a liver-specific enhancer. And so that's what I mean by regulatory activities. Can we actually look through the genome and annotate the genome at a very fine resolution with what it actually is, right? So obviously, some of the things we already know very well from a huge amount of study in biology, right, like promoters. But can we actually annotate, for example, all of the tissue-specific enhancers at the level that we weren't able to do before? And so that's what we started with this model and set out to do. So SAY is a sequence model that I'll show you is much more powerful than that original model from 2015 that I just showed you the architecture for. And what we do is we actually go through the whole genome. At the time that this was published, that was the first time we've been able to do the whole genome. We do go in 100 base pair jumps. Even that took many, many, many months on huge GPU clusters. But now you're going through the whole genome. So since it's 100 base pair jumps, you're going at basically ending up with 30 million sequences, right? Tiling the whole genome. And for each of those sequences, what you're going to do is predict. Remember, so the model was trained to predict 22,000 chromatin profiles. So for each of those sequences, you have a vector of 22,000 long of all of these different chromatin factors, right? From transcription factors to various histone marks, et cetera. And it includes 1,300 cell types and tissues, about 1,000 transcription factors, 77 histone marks. So this is trained on CystromeDB, which is a wonderful resource that really collects all of basically published epigenetic data. The average AUC is about 0.97, so it's quite good at being able to learn these. But here's what we do next. So remember, I promised you regulatory activities. So then what you can do is you can cluster these groups. And you find these very coherent patterns, and we call them sequence classes. So these are really the regulatory. So this is your computational cheap-seq, and I will explain why we actually want a computational cheap-seq when we already have a real cheap-seq. But these clusters are providing this higher level interpretation of what it actually means. So for example, you have classes like FOXA1, ESR1, AR sequence class that's very consistent, or you can have, for example, things like tissue-specific enhancers. So here's a liver enhancer class. What's important to note is we could have done this without any deep learning model, right? Like this is, you could have just taken cheap-seq data from CystromeDB and clustered it. Deep learner was already useful just in terms of cleanup and making this consistent. But that part you could do without the deep learning model. So this is what you get out of that clustering. I'm sorry, with the lights you can't quite see it, but this is the 30 million sequences. Each sequence now is a dot. And the color is this cluster, right? So these are those sequence classes, and you can see there were sequence classes that you saw before, but why we actually need the deep learner and not just the data originally is because now you can actually say, what would a variant do? Right? So with cheap-seq data, no one did, but you could have clustered the cheap-seq data and gotten those sequence classes out of that. But with the deep learning model, now you can say for any variant, what would its effect be? And not simply can you now say, oh, it's going to prevent GATA1 binding, but you can actually have a regulatory interpretation saying this particular A to G change is going to move this sequence from being a liver enhancer sequence to the low activity sequence class. So it's basically going to break down that liver enhancer function. So that's why it's really important. And this is what the SAY architecture actually looks like. So because SAY at the time of training could encompass all publicly available cheap-seq profiles, so about 22,000 of them, essentially, and going around about 4,000 base sequence length, it just ended up being a very heavy computational task. And so a lot of the architecture actually changes from the previous CNN are really about making things faster and better. So for example, there's residual connections in there and dilated convolutions for those of you who are into deep learning. Of course, they're there just for training efficiency. And in fact, it does also actually improve accuracy compared to the original DeepC. There's actually a consistent improvement. But I think the most important part here is really just the whole genome coverage and ability to predict these regulatory classes. It's quite accurate across all of these different sequence types, or sorry, feature types. But I wanted to really focus on what we can actually do with it. So what we can do with it, and I apologize, this is a very colorful slide, but bear with me. It's a little bit, takes a moment to digest. So what we're doing here is we're saying, OK, what can we use this model for? Well, one very clear application is we can actually look at variants that already are known. Right? So these are known disease mutations. And we can try to see, can we actually give a specific mechanism for each one of those mutations? So this is what we're doing. We're looking at all of these known regulatory disease mutations. Here, we're annotating the different sequence classes, right? So this is the promoter sequence class. These are the different enhancers. They're labeled over here, right? So there's the stem cell enhancer, TN1, transcription, right? Erythroblast-like, et cetera. If the variants are sort of within this part of the circle that they're not really sort of strong enough in terms of their perturbation, if they're going out either this way or that way, that means that they're disrupting that particular sequence class significantly, either towards enhancing that class function, right? So making it more enhancer-like, for example, if it's an enhancer, or less enhancer-like, right? So, As you can imagine, more of them end up here. So this is actually disrupting it in terms of making it less promoter-like, for example. But there's certainly some that are actually highly enhancing the promoter. And these are probably, for example, the melanoma TERT promoter, right? Like is not surprising to see here, and that is known. All right. So for example, there's retinoblastoma somewhere over here. Oh, here. These guys are interesting, right? So there's the polydectomy syndrome variants that are actually disrupting this stem cell specific enhancer. And in fact, that is consistent with what their function is in terms of really disrupting the known distal enhancer of sonic hedgehog. And in fact, that mechanism wasn't known, but once we actually predicted this, it totally makes sense and is consistent with what you would expect with the disruption of sonic hedgehog enhancer. And I think I told you about the TERT promoter already. All right, so we can go further with this, right? We can combine this with analysis of common germline variants, right? So these sequence class annotations are actually very convenient for these analyses because they're actually non-overlapping by definition. And so they actually are very powerful in terms of being able to partition the regulatory architecture of disease. And that means that we can actually take them and put them into LD score regression algorithms. And that will allow us to estimate the proportion of heritability that is explained in well-powered different GWAS studies. So in this case, I'm focusing on breast and prostate cancer, so hormone-driven cancer GWAS studies using LD score regression. And you can see that in both of those, right, we are seeing these FOXA1, ARSR1 sequence class. We see some of the promoter sequence classes and several other specific enhancer classes that are coming out. And it's very interesting because there's very consistent germline variant hereditary signature in here between the two cancer types. And then there's very strong differences as well between them. So you can really, and you can do this obviously not just for the cancer GWASs, but for any well-powered GWAS with these non-overlapping annotation. So can we actually go beyond epigenetics, right? Can we go all the way from sequence to expression? So can we actually go to the point where we can take a variant in the sequence and predict what would perturbation be in expression of a given gene in a specific cell type, purely based on variants? So the extreme case of this, and we're doing this actually right now, is if you give me a Neanderthal genome, can I predict what the RNA-seq in Neanderthal would be in different tissues and cell types? I actually have a rotation student doing that right now. And we can talk about what you can do once you have that. But in this case, we're not going to do this on Neanderthals. We're going to take this human genome, right? So remember, we just talked about this part, right? So we can go from the genome through our SAFE framework, and that gives us basically this cheap-seq pattern, right? Now, what we can do is actually now do a supervised training, and we do this with the simplest regularized linear models. Obviously, you could do this with other classifiers. We specifically try to do the simplest possible classifier to minimize the possibility of overfitting. So this is just regularized linear models, and we train it with single-cell RNA-seq. We actually take clustered single-cell RNA-seq. It could be single nuclear RNA-seq. We've tried it. It works perfectly fine. We do not do any reanalysis of these. So the cell type specific clusters really come from whatever data set is providing them. And now what you just have is a mapping all the way from the sequence to gene expression, right? Without having any training of matched genome and gene expression, which is important, again, because if you train on genomes matched to gene expression, you're going to, again, learn common variants. Now, we can talk, and in fact, I'd be very happy to talk about the fact that you can actually do something in between, and you can actually go further in terms of bringing in some personalized genomes, and that's very interesting. But if you do train just, if you take a genome and you match gene expression to it and you train it, it's not just an issue of data availability. You are going to learn, by statistical point of this, really common variants, and we really want to learn the full mapping. All right, so let's see how this works. So can we do this? In fact, we can. So we've done this. So in this is what was published. We actually have a number of other organs as well, but we have this here for kidney, lung, brain, liver, heart, retina, and pancreas. On the left side, you see the number of cell types. Again, the number is not ours. This is whatever was the, the kidney single-cell RNA-seq data set that we used for training. That's what they defined, and we just used that. You can see here AUC-ROC scores for predicting basically highest and lowest expressed genes. So we're really trying to focus on the genes that are actually changing in cell types. So, because you can actually, with these sort of predictors, you can do really well if you just predict something average for everything, right? Like, because essentially there's a huge number of genes, don't change all that much across cell types, and so you can do so well on them that you'll do something extremely boring, but it will look really good. So we're really focused on genes that were changing across cells, and then focusing on them, you still do a very, very good job. Obviously, there's some differences here. I have to say, I would not view this as brain is inherently harder. My gut feeling is, with some evidence for it, it's more data set related versus some inherent feature of gene expression programs related, though it's probably some combination of the two. But these differences are really probably much more data set related than anything else. So what can we do with this? Hopefully, I've convinced you that it works. Now, can we do something useful? So the first aspect of this that we wanted to do is actually annotate some clinical variants. And of course, ClinVar is a database that has clinical variants that was actually sequenced in clinical laboratories. And this is the regulatory non-coding ClinVar variants. And you can say that a huge fraction of this, almost half, are variants of uncertain significance, so VUSs. So basically, somebody came in, they're very sick, there's strong suspicion that there's some genetics that's important. And we don't actually, in here, we're not going to differentiate what the disease necessarily was. We don't know this necessarily for all the variants. For some of them, we do. But the point is, for 44% of the cases, basically, the answer was, we don't know. Right? Only 1% actually come back as pathogenic. So this is a huge problem. And so what we wanted to do is actually use this approach to prioritize clinical VUSs of high functional impact. And remember, you also get a mechanism, right? Because we actually get the specific epigenetic perturbation and then specific expression difference in specific cell type that potentially could have caused this. And I want to show you one. So it turns out if you do that actually for about 300 VUSs, we get a very high confidence prediction that shows that they are perturbed by a score that we actually define from experiments. So we basically, we can do cell culture experiments where we do multiple runs of these experiments, and we can calculate the actual noise level experimentally and link that back to our score differences, right? And that's how we set the cutoff. So it's a very strict cutoff of really experimentally defined. And at that very strict cutoff, we still get a little under 300 variants that we can confidently say perturb gene expression. I wanted to focus on P10 here. P10, of course, is a very important gene in cancer. I don't know if any of you study autism, but it also turns out to be an extremely important player in autism as well. And so we were actually interested in it from the autism perspective. Incidentally, there's actually quite a few cancer pathways that also play a role in autism, which is very interesting. But anyway, here we were interested in P10. And so this is what we saw for P10. We saw that there were a number of VUSs. So on the x-axis is a very approximate sort of version of the genome sequence, right? P10 is somewhere here, right? Here we're looking upstream. Each of those dots is one of the VUSs that was in ClinVar. The y-axis is our model's predicted disruption in expression. And you can see, and it's actually specific to neuronal stem cells because we were interested in autism. That was the model that was closest to basically what we would be interested in for autism. You can see that a bunch of these variants really are not causing much of an impact. But you can see that there is 4, maybe 5, 4 for sure, that are causing a major perturbation, predicted perturbation and impact, specifically in neuronal stem cells. And in fact, what you can do out of these deep learning models is you can do what we call in silico mutagenesis. You can basically feed the model every possible version of the sequence and read out its predicted effect, right? So, and it turns out if you do that, you can literally read out sequence models, right? And so when you read out the sequence model, it turns out to be a YY1 binding site. So that's what's being disrupted. We actually experimentally tested this, and that is indeed what's being disrupted by these variants. And we, in fact, tested all 4 of these variants. I'll just show you one. But in tissue culture with transcriptional reporter assay, specifically neuroblastoma cells, which are reasonably close to the idea of neuronal stem cells, although we are working on trying to get the actually neuronal stem cells and neurons to work in the lab. But here in the transcriptional reporter assay, you get a significant difference in expression based on that single allele change. And we see the same exact picture for all 4. I believe, actually, we did test this one. I apologize, I don't remember. Certainly all 4 of those. And in fact, it is cell type specific. So if we look at, like, for example, HAPG2 cells that are liver cell line, you do not see this significant difference. So you can go all the way from a variant to a predicted mechanism, right? So disruption of YY1 binding site, to a predicted transcriptional effect. So in this case, neuronal stem cell specific disruption of expression. And you can test it, and that's actually the case for a clinical variant. Now, can we actually analyze large numbers of variants with this? What we were interested in is actually, we've actually used these quite systematically, and I'm happy to tell you guys if you're interested in, for example, autism. I pulled this out because that has been published for a while, but we've systematically analyzed the non-coding variants in autistic kids and their unaffected siblings, and showed actually for the first time originally that there was a significant non-coding effect linked to autism that you don't see if you just count variants, and you only see if you look at their disruptive ability, right? So how much they disrupt regulatory function. And we've actually looked now at the expression effects of this as well. Here I'm going to focus on cancer because this is unpublished, and I wanted to share some of the more interesting things in this. And I know there's a lot of interest in cancer here. So the first, this is really more of a sanity check, although I do think it's actually pretty exciting, right? So the question is, if there's really a regulatory signal in cancer, right, can we look at variants upstream of cancer genes? And in this case, I'm using just cancer census, tier 1, very high confidence genes versus tier 2, which are looser, versus non-cancer genes, right, like basically everything else, If there's really regulatory signal that's specific, what we would expect is there would be a different level of disruption that we would expect upstream of each of those in cancer genomes. And so it turns out when we look at this, indeed we see this. So there's a significant difference between non-cancer genes, cancer genes in tier 2, right? So the noisier set, as well as these very high confident genes. So this is, hopefully not super surprising, but it's made us feel good that this is real. Now, can we see anything that's actually specific to a particular cancer? So if we actually focus on a particular type of cancer, so in this case, we're looking at breast cancer, we again see the signal. You can see that obviously we get much higher error bars because we're looking at smaller sets. And in fact, this is specific to breast cancer. So if you look at non-breast cancer genes, you will see a significance in here as well. Now, can we actually go beyond that? And this is all somatic mutations, I should clarify. Can we actually now use this to really look at functional SNPs to analyze GWAS intervals, right? So, of course, I've kept talking about how we don't want to just look at common variants, but of course, common variants are very important. And of course, when we're looking at aspects of quantitative genetic studies, there's always the question of LD blocks and trying to understand which variant is actually the functional one. And so one of the ways that we've been using these methods quite a bit is just trying to see if we have a mechanistic insight in what is the best candidate in these blocks for the functional variant. So I just want to show you an example here of such a question, right? So here we're actually looking at BRCA2 risk interval. And here is from a large GWAS, and I apologize, I don't know where I had a reference. I will dig it up, sorry. But here's the lead SNPs from the study. And you can see that the predicted max epigenetic effect is pretty much within noise, right? Like it's really not predicted to disrupt much. But there is this top predicted SNP that actually is very high. And in fact, it is actually looking real in the GWAS interval, just not as high as these top SNPs. And in fact, actually, if you look at that particular SNP, it's actually within a KB of epithelial expression QTL. So, and the GWAS lead SNP has nothing really around it that would indicate. So, you know, not a proof. We've done this actually for a number of immune disease SNPs with a previous version of our expression prediction model that wasn't cell type specific expecto. And there we actually did test them experimentally and showed that our SNPs were perturbing expression, whereas the GWAS lead SNPs in all of those cases weren't. In this case, you know, I'm just showing you this intriguing possibility. But the point is now you actually do have a way to look potentially at the mechanism of these disruptions and be able to really identify potential candidate SNPs in these regions. And I think the real proof is actually if we can go all the way to phenotypes. So this is actually looking, so this is hot off the press, again, not published. So looking again at somatic variants. And I'm actually looking across the entire ICGC cohort. So this is pan-cancer. Unfortunately, the ICGC cohort doesn't have enough samples where we have survival and whole genomes to be able to link it within cancer. What all I can promise you is that there's neither of those curves just has the whole of one type of cancer, if that makes sense. So this is not just differences in cancer survival by cancer type, nor is it explained by tumor mutation burden. So the only way we're subdividing these patients is by looking at one line that has no high predicted non-coding impact SNPs, and the other one that has a high predicted non-coding impact. And you can see that there is significant and actually quite substantial difference in survival. You know, I'm going to leave you there. We have actually used these models quite extensively for autism, and this is with rare with rare de novo variants. We've looked at across different neuropsychiatric conditions, actually at the level of post-transcriptional variation with common inherited variants. And we've looked actually at rare variants in the congenital heart disease. And in all of those cases, we were able to show very significant non-coding variant impact, in some cases going all the way to phenotypes. Like, for example, in autism, the variants, the higher level of regulatory disruption at the post-transcriptional level specifically is significantly connected to lower proband IQ. So, and you know, autism is highly heterogeneous, so being able to go all the way to phenotype is actually quite remarkable. So non-coding impact is real. It really is playing a role in all these diseases, and that we need to really keep thinking about how to look at its genetic and molecular biology. But I wanted to switch gears a little bit and talk, so a lot of my lab is thinking about these models, right? Like how do we go from the genome to the networks? And then the other side of my lab works the other way, right? Like how do we look at molecular multi-omic data, everything. So I won't talk about spatial here, but we look a lot actually about spatial, transcriptomics, metabolomics, proteomics. In this case, I wanted to talk about multi-omic data and how do we go from these data sets to really building the model of these networks. And of course, the way that I'm hoping will happen is we're trying to bring those two together so we can really start interpreting these variants in their complete cellular and organismal context. So in this case, what we wanted to do is map regulatory circuits from single-cell multi-omics data and specifically focusing on identifying which circuits were dysregulated in particular conditions. So obviously, in this case, we're really focusing on disease, but you can imagine any conditions. And the other point that we really wanted to focus on is thinking about both distal and proximal regulation. So what do I mean? So we have a multiome, and the method, by the way, works whether it's a true multiome data set, right, where we have single-cell RNA-seq and single-cell attack from the same cell, or whether it's parallel multiome, where basically you have the same type of sample. And it's quite robust in that case, right? So we're getting transcriptional regulatory activity and chromatin site binding. So this is really just definitions of what we're looking at from attack. And of course, we're getting gene expression, right, from single-cell RNA-seq. And to be able to take into account the distal regulation, we bring in looping data. So we actually use high-C data. And the really surprising, well, at least it was surprising to me, it turns out that even if you have high-C data that's actually not specific to the particular cell type you're studying, it still helps. I was actually kind of expecting more of a, it would only help if it's really, but it turns out it still helps. You get significant improvement in performance. But of course, if you have high-C data that's actually relevant, that helps a lot more. So what we're going to do is basically build a Bayesian model that's going to integrate all of this to be able to build these sort of circuits, right? So what we want to do is, we're putting 3D genome with a tag and RNA-seq, and we want to identify circuits where, we have distal regulation as well as proximal regulation that's affecting gene expression. And what we're specifically looking for is which ones of those circuits are significantly different between health and disease or various types of disease conditions, right? Resistance, non-resistors, surviving, not survival, treatment response, no response, et cetera. So I will just show you one example of applying this. We've actually extensively evaluated this, including experimentally. But I just wanted you to show one clinical application. So we were looking at a data set. This is in very close collaboration. I think the collaboration acknowledgement is on the next slide. I'll just mention with Stuart Silfon's group at Mount Sinai. And what we were looking at was staph infections. And it turns out, and I have to say, I was actually surprised, although once I found out, it made total sense. So staph infection is a huge problem in the hospitals that I knew. What I didn't realize is that there is really no way to tell if staph infection is antibiotic resistant or sensitive, right? And of course, MRSA is a huge problem in hospitals, right? These antibiotic-resistant bugs are a huge problem. They're very dangerous. And the only way to tell is you just grow them out and see if they're antibiotic-resistant, which, of course, takes a very long time. And so what you end up doing is just treating everyone with the heavy-duty antibiotic cocktails until you find out that maybe they're not actually resistant, which we all can tell why this is a problem. And it turns out if you do what we would all do, which is just gene expression, RNA signatures, it doesn't work. It's exactly random performance, right? So there's no approach that works, and gene expression gives you random performance. If you then do this with these circuits, right? So in this case, we have healthy controls. We have MSSA patients and MRSA patients. We have single-cell RNA-seq and ATAC-seq data that was produced by our collaborators, also Woods and Flowers lab at Duke University School of Medicine as well. And we can put those all in Magical. And it turns out that what we identify is actually a specific interferon-associated circuit. And there's a number of them. And the strongest one actually was in this CD14 monocytes, but there were actually a number of immune cell-specific circuits that we found that were significantly different. So we're not looking at healthy versus disease. That we can do too, and that's, of course, a much easier problem. And that gene expression signature could solve as well. But this is looking specifically at antibiotic-resistant versus sensitive, based purely not looking at the bugs. We're just looking at the patient immune response, right, and their PBMCs. And here's what this looks like. So these are the circuit genes. I believe there are 54 different circuits across all of these cell types that we have significant disruption in circuits. And then we actually looked at, so this was this single cell data set. So not simply did we test this on our test holdout sample, but then we actually looked at 3 validation data sets, which I believe, I don't remember if all of them were microarrays or one was RNA-seq and others were microarrays. So this is very different technologies, totally different data sets, completely in different places. And it turns out that you actually get a pretty reasonable performance around 0.7. So some better, some worse. In fact, the worst one is that microarray data set. But you actually get performance prediction ability by looking at the target genes of these transcription factors in these magic regulated circuits. So you can really get actually an antibody sensitivity prediction out of these magical circuits that you haven't been able to do even if you just looked at the gene expression signature from the same exact data set. And hopefully that gives you an idea of how we can go from sequence and variants all the way to trying to understand their mechanisms and their effects in different tissues, cell types, conditions, and diseases. And then on the flip side, how we can actually start using multi-omic data to map regulation in different health and disease conditions. We also believe in actually making all of these methods available. So we build large frameworks like Human-Base, and actually everything I've talked about is available through Human-Base. I think Magical is available, but not from Human-Base, I should say. That's a GitHub repository. But you can download a number of our tissue and cell type specific network predictions. This I didn't talk about, but you can use Magical to actually Human-Base to understand for any gene set what is really the underlying modular mechanism, right? So for example, if you did a screen and you're trying to understand what's actually going on there, and maybe you did your gene expression or your Go enrichment analysis and it doesn't make sense and you want something finer grained, this will give you these modules, and then you can do the process analysis in these specific modules in its tissue and cell type specific way. And then you can actually do say analysis. So in fact, this is done through the Flatiron and Simons Foundation. So we actually pay for the deep learning cost, right? Like we're not going to be able to do your entire genomes for you, but we're at the Broad, so it's probably not as big of an issue for you guys. But we do really believe in democratizing this, and so being able to allow labs that might not have access to GPUs, either because it's just too complicated or because they don't have the ability to use them, ability to really run these analyses. And this is the say, and this is actually this single cell expector. Sorry, we changed the name. So it used to be Clever. It's ExpectoSC now. All right. So to summarize, I really wanted to just give you a little bit of a summary of what, the way that I really think about what we're doing. And so, and the sort of questions that my group is really interested in, we really think about AI-driven genome scale context-specific view of human biology and disease, right? So can we go from the genome all the way to really molecular understanding of different cell types in different specific contexts, be it developmental stage context, aging, or environmental effects, and bring this all to not just understanding the molecular biology of this, but really trying to link it to the human phenotypes, especially disease-related, but also really a lot about how that affects pathway regulation, as well as cell-cell communication, which I haven't been able to talk to you about, but very interested in, using a lot of mix of technologies, so multi-omic, single cell, and spatial data. This is all, I'm sure, familiar to all of you guys. We're really interested in building integrative approaches, right? And by integrative, I mean both thinking across data types, just computationally integrative, right? So be it genomic, molecular, proteomic, different biomolecules, right? But also across different scales, like how can we build models that really encompass the changes that we see at the cell level with cell-cell communication level all the way to organs, and then finally really at being able to understand how that has an effect on organismal level. And of course, you can see that that's going to be, requires to actually not just build models that integrate more data, but potentially really thinking about very different type of models, right? Like you have to start thinking about pharmacokinetics, pharmacodynamics. There's really a totally different type of modeling. So as much as I would love to say AI is going to be the end-all, be-all, I'm going to predict that we're really going to need to have combinations of machine learning approaches with mechanistic models that are going to be able to really understand what's happening in our bodies and integrate environmental effects. And that's what I really mean by integration of machine learning and mechanistic models. We want to study dynamics and understand effects of various perturbations. And I do have a huge side interest in really thinking how we can integrate data and knowledge across organisms, right? Like, so I am a big believer that as much as I think AI is a huge player in this, I do think that model organisms, and by that I mean everything from cell and tissue culture to actual, organisms, are absolutely critical to biology, are not going to go anywhere in the next, I would say certainly decade or two, quite likely more. And so I think understanding in a much more, not just the semantic level, but actually molecular level, how we actually cross these boundaries is going to be really critical. And I've listed some of the disease interests that we have. And then finally, of course, I don't do anything but walk around, like, write grants papers and, talk to really cool people and give these talks. So this work was done by really amazing students and postdocs in the group. I think they were mostly showing up throughout the talk, their faces, but Chandra Tisfeld and Shi Chen Chandra is actually part, she's the experimentalist research scientist who did all the experiments, but also is part of pretty much every project in the lab. She's really a deep thinking biologist that's, it's been invaluable to have her in the group. She is the lead on the Magical project. Chris Park developed the initial the initial model for predicting post-transcriptional effects of the data. Somewhere I have Kathy Chen developed SAY together with Jian Zhou, who is now a professor, who was my student and postdoc, but now a professor at UT Southwestern, who, by the way, completely independent from me, has, I think, a really amazing model for going from sequence to 3D genome structure that, I mean, we had nothing to do with the model, but we've started using it, and we've been very, very impressed with how well it works. So I'll just tell you if you're interested in that. That's a really good one to look at. And I think Ksenia Sokolova was the lead on the Expecto-SC, the single cell expression prediction model. So I think I've acknowledged everyone, and thank you so much, guys.
Thank you very, very much for a really wonderful talk, Olga. So, questions?
Very impressive talk, Olga. For biologists who study gene regulation, we know that gene expression is a function of like the sequence and the proteins that act on top of the sequence, and of course the chromatin background. So, but obviously from the sequence-based models, it captures some of the cell type specificity. So could you please comment on where this information was captured in the model and how this information are generally applicable across the biological meaning of cell type specific gene regulation?
Yeah, that's a great point. So I'll just back up a little bit because I'm assuming probably there's some computer science. So I'll just back up a little bit on, right? Like, so, I mean, I think one of the most fascinating points in biology is the fact that, right, like we start with basically a single gene, well, okay, two genes, but you know, a cell that has one double copy of the genome. And then we end up with this huge diversity, right? Like I have, you know, huge number of neurons. I mean, if we just look at immune system, there's like who knows how many cell types of just immune cells and counting, right? So how is it that we have the same, more or less, genome and so many different cell types? And that's where the regulation is really coming from. And so that's where the question is a really deep one. You know, how is it our model is just looking at the sequence and yet it's able to generate cell type specific prediction? And the reason is that there is cell type specific cheap-seq experiment, right? Like, so just to be clear, AI isn't, you know, we're not making this up, right? Like this is learning obviously on the data sets that are generated by some of them here, right? But by a number of labs that are generating these cheap-seq experiments. What the models are giving us is now we don't have to generate cheap-seq for every single person with every single possible variant of the genome. But the cell type specific aspects of it are being captured. Now, obviously, most of these cheap-seq data sets are going to be cell lines, right? Like, so there's a few of them that are tissue types, but the vast majority of cell lines. So I will let you make conclusions out of it, right? Like, it's not as bad of a situation. OK, now I'm hand waving, although we do have evaluations on specific, right, like patient effects. So clearly, it's enough to get some clinical predictions. My guess is it's not as bad of a situation as for some questions where cell lines are really not a great situation, because we are hoping that those cell lines actually still have the general epigenetics programs still work, right? Like, so the problem will be if those cell lines are so screwed up that they're really breaking not just the specific, you know, the transcription factor doesn't bind there anymore, but really the whole process of how things are regulated. But, you know, they're a mess. They're cancer cell lines. So, and of course, we don't have as many cell type specific, you know, cheap-seq data sets as we'd love. So as more is generated, these models will get better and better. Right now, they capture some cell type specificity, but not all at that, you know, that first epigenetic. The second one, where we get the expression, that's a bit easier, right? Because now you have these cheap-seq data sets that you predicted, which, as we've discussed, have some cell type specificity, but not perfect. But now you're mapping that to single-cell RNA-seq data, right? And so what it's going to learn is which of those cheap-seq signals are relevant to that specific cell type, right? Like, so let's say CD4 positive T cells, or whatever it is, right? Like, or kidney podocytes that are specifically in a particular activated domain, right? Whatever that single cell data set defined. And the point is, even if the, you know, cheap-seq data sets aren't perfectly reflective of particular cell types, it likely, and we see that, do have enough signal to be able to give you good predictions. But it's not all. And of course, the environment isn't there at all, right? Like, it's completely not there. So we're clearly missing a huge fraction there as well. Does that give a good intuition of? Yes. OK. We can discuss more. All right.
Thanks for the really nice talk. I had a question about an earlier point you made, kind of related to also this previous question, which was you kind of said it might not be a great idea to train models where you have individual genomes and they're matching gene expression or chromatin accessibility. And there were, I think, like a pair of papers last year from Sarah Mustafavi and another person where they actually, measured got some genomes and the gene expression, and then they tried to run it through Informer. And often the sign of the gene expression change relative to the reference was incorrect. So can you just comment on your point there, and what are the right data sets to collect?
Exactly. So that's a really good point. And so that was my second point, saying that it's probably some mixes needed, right? So what I mean is, if we just purely train them on variant information, right, which is basically, which is what the matched genome and expression is, if we purely train on that, we're going to largely learn. We're going to really heavily bias our predictions towards common variants, right? Because that's where we will have, right? Like, these are machine learners. They're going to learn where the signal is strongest, and the strongest signal will be on common variants because we'll see it over and over and over again. Now, that doesn't mean, and we do see that actually, if we look at these models that are not trained with any variants, they do have predictive ability for rare variants, although they do make mistakes sometimes. Now, the question is, the flip side of this, right? Like, so I actually really like Sarah's paper, and I think it's true that they make mistakes. The flip side of it is, if you train purely on taking the common variants, right? The flip side will still be true, that you're not going to learn much about the rare variants. So I think what actually lies as the answer is something in between. We really need models that are trained without variants, but also then either fine-tuned or very carefully optimized to give the personalized genome information as well, right? So I think that's what really is the answer, and I expect that this is going to be coming very, very soon.
I know there are more questions, but maybe we'll stop it here, and then you can go and have extra questions. So thank you very much, Olga.